All right. So I think tomorrow is it you have the placement drive?
Yes, sir.
Okay. So and I think Srinita ma'am told me to wind it by 7:20. So I think let us not spend much time. And maybe we'll check out what to do later on. But for tomorrow, I think we saw some very simple problems of doing GC content and stuff. And I would like to show you what could be the alternative approach if we were trying to use the text files. Okay, so obviously you won't wish to, do everything with short sequences. Maybe you will have to work with longer sequences. Maybe that is sent to you in a faster file, or maybe you'll have to retrieve something. Okay, so we don't have much time to talk about retrieval, but I will try to show you some exercises in which we deal with file handling over here. Okay, so let me try to take you back to one of the exercises we did last class. We did the translation. Okay, that is given. I think we didn't do translation. We did reverse complement, right? We did reverse complement where we given the particular DNA sequence. We did calculate the complement and then reversed it. So if you were to do it on a faster file, okay. So let me take you to that situation here. Okay, so unlike the previous cases where we were, trying to do it using what you can say using the Google Colab, this particular exercise we'll have to do it using the Visual Studio Code. And I hope you are able to see my code. Is it visible to you? Is the code visible? If not, then I'll try another approach. Is the code visible to you on my Visual Studio Code screen? Okay, great. All right. So here again, what I have tried to do is we are trying to calculate the reverse complement, or basically calculate the complement of the particular sequence. Okay, and if you see the data file for this, we have a particular complete genome of this particular virus. Okay, so ideally you will encounter such situations where you are given the entire sequence, and in this case we are given a faster sequence. So I will go through some particular exercises. Maybe you can just go through this and then let me describe it to you, and maybe then we can discuss how this particular code runs over here. Okay, so in this particular code, if you see, we are having what you can say, we are using the same approach that we were doing in the previous case over here. And in this particular case, if you can see, we are using the make trans. Okay, so make trans, and I am passing two sequences. One is the particular sequence, and sequence 2 is the complementary of that. So you can see for capital A, the complementary is capital T. For capital T, complementary is A. For G, it's C. C, it's C. So I am see, if you can see, I'm using both capital letters and small letters. Okay, so in case the faster sequence that you get is in small letters. So for my purpose, if you can see here, the sequence is in capital letters, but maybe if you're downloading it from some other website, maybe it might be in small letters as well. So we do not know that particular situation. Anyway, so we have created the complementary strands, and then we have fed it to the make trans. Make trans, as you remember, is a particular method that allows you to upload, I mean, like, pass a table, and that table can be used for translating the particular sequence over here, right? Then comes the part where we were talking about the file handling part over here. Okay. So, file handling, I hope at least those of you who are here today, you are aware about the file handling. So, let me just go through the particular file handling process over here. If you can see over here, let's see. Okay. So we have this open, and then we pass the name of the file and as the file handle. So here I'm opening a particular file called gene.txt, which, as you saw, contained the gene sequence, the complete genome of this particular virus, papilloma virus 8. Okay. And what we are doing next is we are opening another file. Now, if you remember, in file handling, we pass the name of the file and the particular mode. Okay, so if the mode is not mentioned, then by default the mode is read. And in this example, the file being opened gene.txt is in the read mode because it is not mentioned here. And we are linking a particular file handle called fin to this gene.txt over here, right? And then what we have over here is we are opening another file, gene_c.txt, which is going to store the complementary strand for this particular sequence over here. Okay, so this is open gene_c.txt, and if you see, the mode is W. W is a write mode. Okay, so I hope you know the difference between the different modes. Read is if a file exists, then it is going to read the file. If in case of write, if the file does not exist, it will create a file, and if it already exists, it is going to override the contents of the previous file, whatever that was. Okay, and we have opened it using the with term over here. So generally, the idea is if you open a file, you must close. Okay, so if I were to use if I were to use another without the with condition, then I could have written open file as this, and then I could have done the entire process. But then when I use it with, then it is going to open the file, and at the end of this particular iteration, it is going to close this particular file as well. Okay, so we are opening the file, and here we are opening the file in write mode. So two files opening, and then we start the process where we read the particular lines in this particular file. So for line in fin, which is the file handle for the gene.txt, so I'm reading it line by line over here. And when I read line by line over here, okay, so obviously you will have to look at the file format that you are working on. And in this particular situation, I am working with a FASTA file. Okay, so if you see, this is a FASTA file, and you know the characteristics of a FASTA file. It begins with a greater than symbol, and then you have some information about the sequence, followed by the actual sequence over here. So what I'm going to check over here is I check whether it starts with the particular greater than symbol or not. Okay, so the idea is over here, I want to create another FASTA file. So this gene_c.txt should be another sequence with a FASTA file, and that's why I want to add whatever line I have in gene.txt, and to that I want to add the word complementary over here. Okay, so if you can see, we check line dot starts with greater than symbol. Okay, and then you see I am modifying the line. So line equals line And this is the subscripting. I think one of you mentioned about subscripting in one of the previous classes. So here we have the subscripting. So basically, the subscripting actually means I want all the contents of the line excluding the last one. Okay, so minus one. So basically, from the beginning, excluding the last one, and I add the complementary. Okay, so I add the word complementary, and then I add the last character. So usually whenever you read a line, with every line at the end of a line there is a new line character. Okay, so what I'm doing is the entire line is taken. I just add the complementary this string over here, and then I add the new line character again, and that is now written to the other file. If you see out is the complementary file, so cout dot write a line. And then if you see, there is this keyword called continue. So just to remind you, in loops, in Python loops, we have two keywords, break and continue. Break takes you out of the loop. Okay, so whatever is the current loop, it takes you out of that, and continue skips that current iteration. So basically, wherever the word continue okay, so wherever the word continue will appear, okay, wherever the word continue appears, then what you would happen is whatever lines are below that will be skipped over here. So in this particular case, it writes this line and then skips this iteration, goes right up to the next line over here. Okay, so obviously in a faster file you will have a single line itself, or if even if there are multiple sequences. Okay, so this particular program will work for that. Okay, even if there are multiple faster sequences within a single file, this should work over here, right? So after I modify the first line, then comes the part where we go with the actual translation. So we have the line dot translate and the translation table which was created in the first line over here. So we take the line that we are reading, the sequence line, and then translate it using the trans table. So it writes it to the this file over here. So if you can see, we have, this is the original sequence, and then if you want, I'll just run this file. Okay, so tomorrow, I don't know what platform you will be using to for your lines over here. Okay, so here it says, all right. So what we'll have to do, one thing you need to be very careful is where we are. Let's see if I can find what are the different lines. Okay, so the problem why it did not execute over here is I am not present inside the data directory. Okay, so let me go into the data directory over here. Let's clear the screen. Okay, and then let's rerun, and now it runs over here. Okay, so I have generated this genec.txt, and if you see the output for this, so the FASTA file line is preserved. Okay, complete genome, and the only part that is added over here is this word complementary. Okay, the word complementary is added, and the complementary for the entire sequence. Okay, so this is AATGA. Okay, so TTACT. So you can see the complementary is calculated. Complementary strand is calculated for the entire genome over here. Okay, so this is just one demonstration. You could do lots of things in this particular situation. This is only calculating the complementary. If I were to ask you to calculate the GC content of this particular sequence, I hope you will be able to do that as well. I hope that will be easy, right? So you just need to skip the first line. Okay, and in each line, okay, in each line you have to count how many A's, how many T's, how many G's, and how many C's. Okay, and then you can finally calculate the GC content if you would like to, right? Any questions you want to ask regarding this program? Anything? Any questions maybe?
Sir, the file we shown there like green.text,
Right.
will it be in our system, or we will upload somewhere?
No, I mean, you see what if you remember what I did in the when I ran it initially, it gave me an error. Okay, so what you can do is wherever your .py file is there, you should put the sequence file there itself. Maybe what I can show you my directory maybe. Let's see. Um, okay. Okay, so just see my directory over here. Um, so this is the directory where I have some py files. So for binc 1.py, this is my file, and in this file itself, I have the FASTA file. So the simplest way out usually is to put the FASTA file within the same directory as the sequence, the Python file itself. All right. That is a simple approach, but then maybe sometimes if you have too many sequences, you'll have to put the sequence file in a sequence directory, and then for the programs they would be in a different directory itself. All right. So it depends upon the situation. Okay, depends upon the situation where you would be asked. Okay, so you don't have to upload it anywhere. Wherever your sequence is there, you put wherever your py file is there, there you put the sequence. Mind you, this is a different approach compared to the last two classes that we did. I am using a IDE called Visual Studio Code. If you can see right on top, this is Visual Studio Code, which actually helps me to run the Python file. Okay, so I write my code and I run my code over here. So this again becomes difficult if you are using Google Colab. Okay, Google Colab, then maybe I'll have to give a link to the Google Drive. Okay, so Google Drive link, but I have not tried it out. I'm just hypothesizing that is going to work. Instead of just writing the name of the file, you might have to paste the link to the file, and then you could try any of these operations. Okay, so I'm not really sure for tomorrow's placement drive how you are expected to write the coding round. I hope it is on some sort of IDE. Okay, so just write your Python file and have your sequence file, whatever they give you. If it is a DNA sequence, fine, or it's a protein sequence, or it could be another FASTA-Q sequence, or anything. Just put it in the same directory as your Python file. Yeah. Anybody has? Yeah. Yes, please.
Sir, can you once again please explain from line number 4?
Line number 4. Were you there for my class yesterday?
Yes, sir.
So this part I hope you know this make trans and then translate,
Yes, sir.
right? So that part you are clear, right? How the translation is happening. Uh, uh,
Yes, sir.
but then are you familiar with file handling?
Sir, actually, the line number 8, that is line, then it is colon, then -1.
Yeah, yeah.
That means, sir, it is for the specific word. I think that we have to minus from the backside of that word. We have
Exactly,
to.
exactly. So, no, so let me
Yes,
show you
sir.
that. So see this one. So basically, this line is only to take the first line over here, right? So this is if this is the sequence, I am beginning from the beginning and going up. Okay, just I scrolled up. Yeah. So from the beginning, I go up till the end till genome E. Okay, I don't go with the last character. Now remember, in any file there will be a new line character for after every line. Okay, so the what I am trying to do over here is I am just trying to copy this line, and to this line I want to add this term called complementary. That is basically what I am trying to do. Just add the word complementary so that this will be visible in the result file. So you can see in the result file I have the same line as was there in the previous case. I've just added the word complementary over here. So this gets added, and then you can see the what you can say the new line character is added over here. So line -1 is the last character, which is the new line character gets added over here.
Okay, sir. Then in line number 10, you have mentioned continue. Sir, can you explain a little bit?
Yeah, so I told you in Python loops you have two words, break and continue. Break is to come out of that particular loop, and in this case I've used continue. So I don't want to stop there. Okay, so I just want to skip whatever lines are present after this line number 10. I want to go for the next iteration. Okay, so if I hit the greater than symbol, I know that is the start of a sequence. So I modify the header of that FASTA file and then continue. So I don't execute this line number 11. I go back to the next line. And the next line, if you see in a FASTA file, what do you have? You have the greater than symbol followed by the sequence header followed by different sequences. So if I hit a sequence, obviously the sequence will not have this greater than symbol, it will be a normal sequence. So for that sequence line, I can do the translate. I can run line.translate, so I get the translate translation. I should not say translate, I mean I get the complement. Okay, I get the complementary sequence, and that I write into the new file.
Okay, sir. Thank you, sir.
Okay. Any other questions? If not, then I'll just discuss another problem before we close. Okay, so I hope there are no more questions. Let me now go to another problem. Let's see if I can open that for you. Okay. Okay, so this is a particular problem. So we want to find the ORFs on the forward strand for the given nucleotide sequence using only the start and end codon patterns. Okay. I hope you are aware about the ORFs, open reading frames. You are aware about that, right? Open reading frames. Just confirm so that I don't have to talk about ORFs.
Yes, sir.
Okay, great. So if you are aware about ORFs, then so basically the idea is I need to go through this particular same gene.txt, so which is this papilloma virus, and I want to take this particular sequence and find out all of the particular open reading frames that I have over here. And how are we going to detect this over here? So I'm going to look at the start and end. Okay, the start codon, end codon, so wherever they appear, I take the entire sequence, and I am only looking at the forward strand. Okay, so if I were looking at the 6 frame translation, then I would have to look at the forward three proteins, 3 protein possible translations, and then the 3 in the next complementary strand as well. Okay, so let's have a look at this particular code and this particular case. So here again, as I was telling you, we have the gene.txt, and I'm trying to choose all of the particular ORFs, which will have minimum of 100. Okay, so the length will be at least 100. So I want to choose those sequences over here. And so first I'll have to look at these ORFs, and for ORFs I'm taking an empty dictionary over here. Okay, and what I'm going to do over here is we look for different frames. In this particular case over here, let's have a look at the gene.txt, right? So first we are going to open this gene.txt, and then I start looking at this particular FASTA file over here. Obviously, if we have this greater than symbol, I am not very interested in the greater than symbol. I'm just storing it in this variable called DESC, which is for I should have made a description or something that makes it easier. So I'm just going to store, if it is the greater than symbol, I'm going to store it as a description over here. And then what I'm trying to do is I'm going to continue. Like in the previous case also, you see, whenever there is a header, I am skipping over, I'm just dealing with the header and then skipping over to the next line. So here also we are skipping over to the next line. And what I am doing is from the next line, or where I get the actual sequence. So from the line number 2 till the end, we hit the particular sequence. So all of these sequences now, I am storing it in a variable called total sequence. So total sequence, if you see, it's a string variable. It is going to contain the entire length or entire sequence. Okay, so if you see, we are doing total sequence plus equal to line colon minus 1. So this line colon minus 1 means we are excluding the new line character. So for the first iteration, it is going to take this entire sequence till G. Okay, not this new line character which is the at the end. So that we are discarding. It takes this line, and then for the next iteration, line number 3 gets added to line number 2. If you remember, when we in the first class we were using this plus equal to, right? So plus equal to stands for, so if you have an existing string in total sequence, we add another string to it over here. So if you remember, total sequence here, we have initialized it as an empty string, and every time we hit a sequence line, we keep adding to the total sequence. So by the time you are done with this loop, okay, so this starting from line number 9, where we begin with the gene file and we are reading the entire sequence. So in the total sequence, now you will have the entire FASTA sequence you'll have inside that over here. Great. So after doing that now, what we are going to do is we are going to do the actual translation, right? So we'll let's look at this particular frame over here. So finding the frame 1. So here I am using a particular function called range. Okay, so range is again a particular function which actually is able to fetch us the particular, the number over here. So we have range 0 till length of total sequence, and we have 3 over here. So what we want is we want the three things over here, right? So as you know, in a codon we have 3 nucleotides, so we're going to take 3 codons over here. So we go from 0 till the total length of the sequence, and we take 3 nucleotides at once. Okay, so that's the number that will be loaded inside n. Okay, so for first iteration it will be 0. Then you can see the codon we are choosing. Codon equals total sequence 0 colon 0 plus 3. So it is 0 colon 3, right? So the sequence at the 0th location, 1 and 2. So that's the first codon over here. And then what I am trying to check is I am to check whether it is the start codon or not. Okay, whether it is the start codon or not. That's what we are seeing. ATG being the start codon. Okay, and if it is ATG and I hit the start codon in the beginning itself, then I'll store whatever is the value of n, that is the starting point of the codon. I'll store that as the start over here. Okay. Now, another situation could be if I am hitting the stop codon over here. Okay. So if I am hitting the stop codon, that is, it could be TAA, TGA, TAG. So this is a particular in operator that we are using. Okay. So we are checking if the codon that we have retrieved in this particular case, whether it belongs to this set. I should not use the word set, but then if it belongs to this particular list, I could use a set, I could use brackets over here to make it into a tuple. Okay, so cordon in either of these situations. Okay, so whether it if it matches over here, then we do the end equals. I mean, we store n as the end over here. Now, as soon as we hit the end, okay, as soon as we hit the end, and if start has been initialized, okay, if start has been initialized, so we're basically going from the beginning till the end. Okay, so if we hit n, and then whether if start is initialized, that's why I've written if start. Okay, so if start has been initialized, then I will take the ORF as start till end plus 3 over here. Okay, start colon end plus 3 over here, and then I check whether the particular what you can say this length. Okay, so whether the length is greater than the minimum length of this ORF. The minimum length we have taken as 100. This is an arbitrary limit. So if it is greater than that, then we are going to do something else. So what we do is I am taking this start till the end. So we are keeping it as the key. Okay, we are storing it as the key in this particular ORF dictionary. Okay, so ORF dictionary, we keep the key and then we try to give the length. Okay, so from this length, this position to this position is a particular ORF over here, right? And then here I come up with the different ORFs over here. Okay, so let's see if what are the ORFs in this particular situation over here. Okay, so it is trying to process that, and you can see for the entire length I have got these ORFs over here. So 243 to 507. Okay, and then from 642 to 765. So this is one ORF. Okay, and then this is another ORF. So different ORFs, or this could be some code for some proteins maybe. So you can show these things over here. Okay, you can show this entire proteins. Okay, and you could combine it with a translate program. So if you have the codon table, okay, so for example, this is the codon table over here. Okay, so sorry. So if we have the different codon tables in this particular case, okay, so you can take a codon table and maybe if you want, you can put it in a dictionary. Okay, already you can store it in a dictionary, and then you could even do a simple translate. Okay, you could do a simple translate of this particular sequence. You could see it over here as well, right? So if there are any questions for this particular program, any questions or anything that you would like to ask? Anything you want to ask regarding this ORF problem? Uh, I mean, like, if there is a question, you ask. Otherwise, um, maybe we can discuss if there is anything. Yeah, yeah.
Can we do all of this program in PyCharm editor?
Yes, yes, very much,
Yeah.
very much. I mean, I am comfortable with VS Code. That's why I'm doing that. PyCharm also the same thing you'll have to do, right? The same code is there. Just write it in PyCharm and make sure the path is correct. Path to your FASTA file. If the path is correct, everything is going to run as it is. I mean, nothing is going to change. Any idea about what how you are going to attempt your code tomorrow? Will you be doing it on your own systems or
So,
yeah, yeah.
so in tomorrow's placement exam session, there is given three rounds: MCQ round, Python and aptitude. And in the
Mm-hmm.
second round, there is given coding assignment round. What does that mean?
Okay, yeah, Srinita did share it with me as well. Just a moment. Mm, okay. So you have the PPT talk and then the MCQ round. Okay, so I think the MCQ round probably consists of two parts, right? Aptitude and Python. So you might have some multiple choice questions on Python. What I would suggest is you could on the web you could go through this W3 schools or you could just some very basic stuff. You could try out those things. And the coding assignment round probably it will be a problem like what I discussed now. Might not be so simple. You might be given a faster file and try those questions itself, GC content and those things can be done, or maybe you might be even given a protein sequence and some properties you might be asked to calculate from the different files. That could be some situations, or even though I am not prepared with anything like that, but you might be even given a PDB file. Okay, so from the PDB file, you might have to extract some sequences, or maybe particular, C alpha atoms or things like that. That also could be part of this particular coding assignment. So I believe you will be given a problem, and then you'll have to code for that.
So in that third round, assignment discussion round and case study, that kind of a question.
Yeah, sorry, could you repeat your question?
We can see that third uh, assignment discussion round
Yeah,
for
yeah,
the coding assignment.
yeah, yeah, yeah. Assignment discussion round. Okay. I'm not really sure about the assignment discussion round. Whether they are talking about particular case study. I mean, they did mention case study. Maybe they might give you some problems regarding certain situations. Maybe from a NGS data set or some particular data set, and then maybe ask you to give your opinion about that. I'm not really sure, I'm just guessing, but at least I'm sure about the MCQ and coding assignment, which is kind of obvious also. Yeah,
Thank you, sir.
right. All right. Is there any more questions? Maybe I think I prepared these two questions, and maybe for your practice, you could try these approaches for uh, uh, your other, whatever problems we did in the previous classes about GC content and other things. You could just try out in that problems as well, and you should also try out with some of the PDB questions. I do have a PDB question, but then I have not given the solution for that as well. So for this example, you read a PDB file from the PDB and then extract all the coordinates of the water oxygen atoms. Okay, so bit of domain knowledge obviously is required, to study what is there in a PDB file, what are the different water molecules. Okay, and specifically they are talking about oxygen in the water molecule HOH, right? So then what you need to do is calculate the distance between all of the oxygen and identify which will form the hydrogen bonding. So this again requires a bit of domain knowledge, right? So you could get problems like this. Okay, so given a particular file, then extract something and calculate something. So that could be some particular work that you could do as well. Okay, so a lot of such problems can be given, right? So again, from the same PDB file, extract all the peptide bonds. Okay, make a list of all of these. Okay, and then calculate the torsion angle for all of these peptides. These sort of simple calculations. So even though these are might not be the case, you might be even given NGS data. Okay, so some sort of analysis on the FASTA-Q files as well. So that could be the possible questions. Maybe very less time we got, but then hopefully you perform well tomorrow. And if required, yeah, yeah,
Sir, I have a question.
yeah,
Sir, for PDB file,
yeah.
we have to download the PyMol app. I mean, in our computer.
See, for the PDB file, if you are visualizing, you need PyMol. Okay, so visualization के लिए PyMol चाहिए for otherwise if because PDB file is a simple text file. Okay, so if you are asked to work on a Python script, then you can directly do it. Just download any PDB file, or you might be given a PDB file. So you need to just open it in a file handle, and then you could work with that. Anything. Because you know the portions. The basic idea over here is you should know different file formats. What is a FASTA file, what is a PDB file, what is a FASTA-Q file, what is a GenBank file. So different file formats again is kind of important over here. And I'm not really sure if they'll ask you anything fetching data online. I'm not really sure of that. But if that is the possibility, then you will actually have to refer to some documentation. It's like NCBI has the e-utils or things like that. Any particular API and stuff. I mean, we don't have much time to discuss, but then that could be the situation. Okay, okay. So, all right. So I think I will stop here, and if required, we'll meet further. I mean, if some of you get through, it will be really great. Um, but if there is any requirement for any other classes for Python and all, we'll continue with some more problems. So it seems you have an exam at 7:30. Do you have an exam at 7:30?
No, sir. So actually, IMTs have exam at 6:30.
Oh, but then, अच्छा.
So 7:30 को था but वो prepone हो गया.
Oh, prepone हो गया है. Okay, okay, okay. All right.
वो 5 हाँ 5 10 को हो गया.
अच्छा, अच्छा, अच्छा. ठीक है, ठीक है. All right then. If that's the case, so I'll just stop here. If you have any questions, I'll answer that. Otherwise, I think I prepared these two questions, and for your purpose, I'll just post it on the Google Classroom. So to check out the code. If
So can you give us example if questions will be in the
see.
questions
See, these questions that I am showing you is from a BINC exam. So I think till 2018, the along with the DBT JRF, there used to be a Bioinformatics National Certification exam. So these were the types of questions we used to get for the coding round. In the sense that was a three-layered exam where first round used to be some normal physics, chemistry, bioinformatics, and stuff like that, and then second round used to be the coding round. Okay, so coding round, and then we had the theory. Okay, so coding and then theory was there. So theory had maths and all. So these are the types of situations we used to get maybe in around 2019 or 18. But if you look at the industry, there could be different requirements. Okay, so within the whatever short time I have, I came up with these situations. But if you look at what sort of questions you might get, um, you could also encounter, maybe we could have these regular expressions. Using the regular expressions, maybe you'll have to find certain patterns. Okay, you might have heard of prosite motives. Okay, so given a prosite motives, if you can try to find out where all, you have a glycolization site or some post translation modification site. So how would you find that out in a protein sequence? So that could be some of the problems that you can get. And then mainly you could also look at analyzing blast, the output of blast, right? So if you run a blast and you are given a blast output, then from there, if you have to extract certain sequences that have bit score above 200 or a particular e value, so extracting those columns and all, even though Python is a overkill for those situations, maybe a bit of shell scripting you should know in Linux and all. So that could be some of the problems I can just think of. Yeah, so have a look at uh, I mean, uh, some of these problems. Maybe concepts are quite important. Um, in the sense how what these files are. If for example, given a PDB file, if you are aware of all of the different parts of the PDB file, you can extract a certain portion, maybe the sequence. Okay, so sequence from the PDB file. So PDB file along with the structure, there is a certain portion where they keep the sequence. So if you are asked to extract the sequence from the PDB file, so you could do such problems, just as a practice to hit that. Anyway, so whatever problems you are given, it will be mainly Python's work is to extract, to analyze. These things can be done. And I rarely think they will ask you to directly work with BioPython, because BioPython again does most of the jobs directly. So you'll have to know how to install BioPython and that. But usually they don't ask BioPython. You might be asked how to use the concepts of Python. BioPython would probably come at a very later stage. Yeah. Okay then. So let us see how all of you perform tomorrow, and then we can plan further classes. I could, talk about some basics, or I'll take up some problems and then try to solve them as well. Right?
Okay, sir.
Okay, then all the best for tomorrow. And I'll see you whenever I'll see you.
Okay.
Okay.